Assalamu alaikum. Good morning, everybody. Today we'll start to talk about the physiology of the cardiovascular system, and we'll start first with the heart. So the title of today's lecture is the properties of cardiac muscle tissue. The objectives are to describe the functional organization of the cardiovascular system, define the terms rhythmicity, excitability, conductivity, and contractility, describe the conduction system of the heart, and outline the normal pathway of the cardiac impulse, describe the action potential and the contraction of the ventricular muscle, then describe the excitation contraction coupling in the cardiac muscles and compare it to excitation contraction coupling in the skeletal muscles, and finally describe the electrical events of a normal ECG and how it is related to contraction and relaxation of the atria and ventricles. As we know from anatomy, the heart is the pump that ejects blood and is responsible for the pressure gradient that is responsible for the flow of blood throughout the cardiovascular system. The heart is composed of 4 chambers, 2 atria and 2 ventricles. The right and left atria, and we have the right and left ventricles. The chambers are separated by interatrial septum and interventricular septum, and also the heart has 4 valves that secure unidirectional blood flow or blood flow in one direction. The thickness of the both ventricles is different. We have a much thicker left ventricle compared to the wall of the right ventricle, and this is because the left ventricle has to eject blood against or into the high resistance systemic circulation, while the right ventricle ejects blood into the low resistance pulmonary circulation. The heart is one of the organs of the cardiovascular system, and as I said, it is the pump that is responsible for the generation of pressure gradient that will secure the blood flow throughout both the systemic and pulmonary circulation. The left ventricle is a strong pump that will pump blood into the systemic circulation, to the skeletal muscles, to the kidneys, digestive tract, brain, and other systemic organs, including the skin. And blood from these tissues or these organs will go back to the right atrium, to the right side of the heart, and then it will flow into the right ventricle. The right ventricle will pump the deoxygenated blood into the pulmonary trunk and its 2 branches, the right and left pulmonary arteries, into the lung, where gas exchange will happen between air in the alveoli and blood in the pulmonary capillaries. Then oxygenated blood will flow back to the left atrium through the 4 pulmonary veins. The heart wall is composed of 3 layers. These 3 layers are from inside to the outside: the endocardium, then the thick myocardium that contains the cardiac muscle cells, and then the pericardium. We can see here in this part of the figure or the slide the 3 layers clearly. We have an endocardium, which is a simple squamous epithelium, and then we have a thick myocardium, and then we have the pericardium, which is also a serous membrane that forms the visceral pericardium. And we know that we know very well that the heart has pericardium composed of parietal layer and visceral layer that is separated by the pericardial space that contains a very thin layer of the pericardial fluid. The myocardium is composed of cardiomyocytes or cardiac muscle cells. The cardiac muscle tissue is the principal tissue of the heart. As we saw in the previous slide, it is the thickest layer of the heart wall. Cardiac muscle fibers have arrangements and striations similar to the skeletal muscle fibers, but they are shorter than skeletal muscle fibers and they exhibit branching. The skeletal muscle fibers are long and they do not branch. As you see here, we have short skeletal muscle fiber, and this skeletal muscle fiber is showing branching or is branching. The cardiac muscle fiber has usually one centrally located nucleus and in few occasions two nuclei. The skeletal muscle fiber, or the one cell of skeletal muscle fiber, contains many nuclei that are peripherally located just beneath the sarcoplasmic membrane. And the T-tubules in the cardiomyocytes are wider compared to the skeletal muscles, and they are less abundant than the skeletal muscle fibers, and you will see how these T-tubules and their shape in the lecture of skeletal muscle fiber in the later part of this course. The sarcoplasmic reticulum that contains calcium, which is important for contraction in both skeletal muscle fibers and cardiac muscle fibers, are smaller in the cardiac muscle fibers compared to the skeletal muscles. You can see here we have the T-tubules that are invaginations from the cell membrane, and we have the sarcoplasmic reticulum, which is smaller compared to the skeletal muscles. And also you can see the striations here and here due to the special arrangement of the contractile proteins, the thin and thick filaments in the sarcomere of the muscle, and you will study these striations and the sarcomere of the muscle and its arrangements in the skeletal muscle lecture. The ends of cardiac muscle fibers connect to neighboring fibers or to adjacent muscle fibers by irregular, as you see here, by irregular transverse thickening of the sarcolemma or the cell membrane of the cardiomyocyte, and this irregular transverse thickening is called the intercalated discs. So this is one intercalated disc, here we have another one, and here we have a third one. If we take a closer view of the intercalated disc, we can see that it contains, or it is very rich in gap junctions. The gap junctions are membrane proteins that connect the cytoplasm of adjacent cells. The intercalated discs contain desmosomes which hold the fibers together, and gap junctions which allow muscle action potentials to conduct from one muscle fiber to its neighbors or to the adjacent muscle fiber. Gap junctions allow the entire myocardium of the atria and the entire myocardium of the ventricles to contract as a single coordinated unit, and we call this the functional syncytium that I will explain in more details later on. Cardiac muscle cells have bigger and much more mitochondria compared to the skeletal muscles because they use mainly aerobic metabolism, or they depend mainly on aerobic metabolism. In a cardiac muscle fiber, mitochondria occupy about 25% of the cytosolic space or the volume of the cell. It is only 2% in the skeletal muscles. Cardiac muscle contraction is about 10 to 15 times longer than the skeletal muscle contraction duration. And you will study the skeletal muscle contraction, as I said, later on in a separate lecture during this course. Cardiac muscle contracts when it is stimulated by its own rhythm. The heart contains specialized cells, we call them the autorhythmic cells, that I will talk about them in details later on in this lecture. There is a concept called, or in physiology we have something called syncytium. We have structural syncytium and functional syncytium. The structural syncytium, when a group of cells will lose the boundary or the cell membrane that separates between them, and they will become a large multinucleated cell. And one of the good examples is the placenta that will be covered in the lecture of pregnancy at the end of this lecture. The heart is a good example of functional syncytium, where the muscle fibers retain their own plasma membrane, but their cytosols are connected with each other via the gap junctions that are present in the intercalated disc. So when one cardiac muscle cell undergoes an action potential, or there is a change in the voltage or the membrane potential of that muscle, the electrical impulse spreads to all other cells that are joined by gap junctions with this cell, so they become excited and contract as one single unit. We call it the functional syncytium. The atria and the ventricles are separated by the fibrous skeleton of the heart. So the atria, or both right and left atria, they will function, or they will contract, or they will get excited and they will contract as one unit. So the atria are one functional syncytium and the ventricles are another functional syncytium. They are electrically isolated except for the AV bundle, the atrioventricular bundle, that we'll study in the coming slides as part of the conduction system of the heart. Here in this table we can see a comparison between the features and functions of the skeletal muscles and cardiac muscles, and as I said, skeletal muscle fibers will be covered in more details in one of the lectures before the end of this course. So in terms of structure, skeletal muscles are striated, long, cylindrical, multinucleated. The cardiac muscles are striated, short, branched, and they contain in most cases one nucleus, and in few cells we have two nuclei. Skeletal muscle fibers are not connected by gap junctions, but cardiac muscle fibers are connected by gap junctions at the intercalated disc. In terms of their contraction as a single unit, since there are no gap junctions between skeletal muscle fibers, they do not contract as one unit, they will contract individually. In case of the cardiac muscle, because they have gap junction, they will contract as one unit, and this is called functional syncytium. T-tubules are abundant in the skeletal muscles, but they are few and wider in the cardiac muscles. Sarcoplasmic reticulum is extensive and has terminal cisterns. In the heart or the cardiac muscle it is less extensive and there are no terminal cisterns. The source of calcium for contraction in the skeletal muscle is coming only from the sarcoplasmic reticulum, that is extensive and more developed in the skeletal muscle. In the case of the heart, it comes from both the sarcoplasmic reticulum and the extracellular fluid. Okay, these are the main differences. Now, what are the properties of cardiac muscle fibers, or the cardiac muscle in general, or cardiac muscle tissue? The cardiac muscle tissue has four main properties. Number one, they exhibit something called autorhythmicity. They can generate their own rhythm of action potential. It is the ability of the heart to beat continuously due to an inherent and rhythmical electrical activity. Number two, excitability is the ability of the cardiac muscle to respond to a stimulus of adequate strength and duration by generating an action potential. Conductivity, it is the property by which excitation is conducted through the cardiac tissue, and this feature is mainly dependent on the gap junction that is present at the intercalated disc. Contractility is the ability to contract when stimulated. What are the autorhythmic fibers that form the conduction system of the heart? Some students think that the autorhythmic fibers that are responsible for the stimulation of the heart and the contraction of the heart, or the generation of action potential, are neurons, and this is the wrong concept. The autorhythmic fibers are specialized cardiac muscle fibers, so they are cardiomyocytes. They are a special type of cardiomyocytes. Autorhythmicity is an inherent and rhythmical electrical activity, or an inherent and rhythmical electrical activity is the reason for the heart's lifelong beat. The source of this electrical activity is a network of specialized cardiac muscle fibers. I repeat again, the autorhythmic fibers are specialized cardiac muscle fibers. They represent 1% of all cardiac muscle fibers that are present in the heart. These specialized cells are called the autorhythmic fibers because they are self-excitable. They can generate action potential without any external stimuli. They repeatedly generate action potentials that trigger heart contractions. So the heart can contract even without the effect of the autonomic nervous system. Two important functions of the autorhythmic fibers. Number one, they act as pacemaker, setting the rhythm of the electrical excitation that causes contraction of the heart. So they are responsible or they generate the heart rate. So they are behind the heart rate. Number two, the autorhythmic fibers also form the conduction system, which is a network of specialized cardiac muscle fibers that provide a path or way for each cycle of cardiac excitation to progress or propagate through the heart. So they are responsible for generating the heart rate and also for propagating the action potential or electrical activity throughout the heart, from the atria to the ventricles. The conduction system of the heart is formed by the special arrangement of the autorhythmic fibers in the different chambers of the heart. As you can see here, we have both atria and we have both ventricles, the right and left ventricle. The first arrangement of the autorhythmic fibers is present, or the first collection of autorhythmic fibers is present in the wall of the right atrium, and this is called the Sinoatrial node, or the SA node. The other, or the next group of autorhythmic fibers are present at the junction between the atria and the ventricles, and that's why we call it the atrioventricular node, or AV node. Then a group of fibers will form a bundle that will connect the atria with the ventricles. As I said, the atria are electrically isolated from the ventricles by the fibrous skeleton of the heart, and this bundle of fibers that is called the atrioventricular bundle is the only electrical connection between the atria and the ventricles. Then the atrioventricular bundle, or the AV bundle, or bundle of His, will give me right and left bundle branches. They will run down the interventricular septum to the apex of the heart, and then they will give me the bundle branches that will supply the left ventricular wall and the right ventricular wall. The autorhythmic fibers do not have stable resting membrane potential. I said these are specialized cardiomyocytes that can generate action potential without any external stimuli. They can generate action potential on their own. they repeatedly depolarize to threshold. This is the line of threshold at the end of an action potential. If you remember from the structure function, action potential is a change in the membrane potential or membrane voltage. It has two parts: depolarization, repolarization, and after hyperpolarization. In the neuron and skeletal muscle fibers, after hyperpolarization, then the muscle or neuron cell membrane will return to the resting state and will remain flat and stable until the next stimulus. However, in the autorhythmic cells, the membrane potential will not remain stable. It will gradually depolarize until it will reach the threshold. When it reaches the threshold, depolarization will occur on its own and the action potential will be self-generating. Okay, so they repeatedly depolarize to threshold spontaneously, and this is why the autorhythmic cells can generate action potential on their own. The spontaneous depolarization, or this part of the action of the change in the membrane potential, is called pacemaker potential. Pacemaker potential is not an action potential. It prepares for action potential. So it is a gradual change in the membrane potential, or gradual or spontaneous depolarization, that brings the membrane potential of cardiomyocyte to threshold, and then an action potential will be triggered. So when the membrane potential reaches the threshold, it triggers an action potential, as we can see here, which has two limbs: depolarization and repolarization. On their own, the autorhythmic fibers in the SA node would initiate an action potential every 0.6 seconds, which means they will generate about 100 action potentials per minute. So the autorhythmic cells in the SA node, and I repeat in the SA node because autorhythmic fibers, they have different rates of action potential generation. So the highest rate of action potential generation is done by the autorhythmic cells that are present in the SA node. So the autorhythmic cells in the SA node, they can generate up to 100 action potentials per minute. This rate is faster than that of any other autorhythmic fiber in the heart, because action potentials from the SA node spread through the conduction system and stimulate other areas before the other areas are able to generate an action potential at their own slow rate. The SA node acts as the natural pacemaker of the heart. So the pacemaker of the heart is the part of the heart that takes the lead in generating and determining the heart rate, and this is, or it lies in the SA node. The rate of generation of action potential is different in different parts of the conduction system. As I said, in the SA node it is 100 action potentials per minute. In the AV node it is between 40 to 60 action potentials per minute, and the AV bundle branches and the Purkinje fibers, it is between 20 to 35 action potentials per minute. It can be up to 40. So different parts of the conduction system, they have different rates of action potential generation. Why the SA node is the pacemaker? Because we said it is the part that fires at higher rate. SA node acts as the heart pacemaker because it has the fastest rate of generating action potential. Just if we compare that or give an example like a train, the first part of the train, as you see here, has the highest speed, so it is the one that determines the speed of the entire train. Okay, the SA node or non-SA nodal tissues are latent pacemakers that can take over at slower rate if the normal pacemaker fails. In some conditions, as you will study in the lecture of arrhythmia in phase 2 during the cardiovascular module, in some diseases the SA node is damaged or fails to generate action potential. So if it will fail, the next car in the train will take the lead, and the speed of the entire train will depend on the speed of the second car. And this is the easiest example to understand why the SA node is the pacemaker of the heart. If the SA node fails, then the AV node will become the pacemaker of the heart. If both SA node and AV node fail, then Purkinje fibers will become the pacemaker of the heart, but the heart rate will be around 30 beats per minute because they have lower rate of generation or lower rate of action potential generation. As I said in the previous slide, that SA node autorhythmic fibers, they can generate 100 action potentials per minute, which means that we have heart rate of 100 beats per minute. But why normal SA node, why the heart rate is 70, why it is not 100? As I said, normal SA node fires at 100 beats per minute or 100 action potentials per minute. However, the heart rate at rest is 70 beats per minute. Why this difference? You will know in the... As I said, that the heart can beat on its own because it contains autorhythmic cells, and we have the autorhythmic fibers in the SA node that can fire at high or at the highest rate, so they are the pacemaker of the heart without the need of the intervention of the sympathetic and parasympathetic system. However, sympathetic and parasympathetic system can influence the firing rate or can change the heart rate, and we have studied in the autonomic nervous system that sympathetic nervous system increases the heart rate and parasympathetic system decreases the heart rate. So if we see or have a look at this part of the figure, the black dashed lines are the action potentials or the action potentials at the normal rate in the SA node. And then if the parasympathetic system is stimulated, the SA node pacemaker activity will decrease because the pacemaker potential will change. It will become less steep, so we'll have action potentials generated at lower rate. On the other hand, the stimulation of the sympathetic nervous system or the effect of sympathetic nervous system stimulation on the SA node will make the pacemaker potential more steep, and this will lead to the generation of more action potential because the membrane potential will reach threshold quickly or in shorter period, so we have more firing, and that's why we have higher rate or high heart rate under sympathetic stimulation. So the pacemaker potentials in the autorhythmic fibers, nerve impulses from autonomic nervous system divisions, and blood-borne hormones like epinephrine from the adrenal medulla modify timing and strength of each heartbeat, but they do not establish the fundamental rhythm. So we need the SA node and the rest of the conduction system to generate the action potential. However, the autonomic nervous system and hormones can change the frequency of heart rate and change the strength of the muscle contraction. In a person at rest, for example, acetylcholine released by the parasympathetic division of the autonomic nervous system slows the SA node pacing to about every 0.8 second, which brings the heart rate down to 75 action potentials per minute or 75 beats per minute. If we cut that parasympathetic supply to the heart, then the heart rate will become 100, which is the firing rate of the autorhythmic fibers in the SA node. Okay, now how the action potential, we said that the SA node will generate the action potential at a frequency of 100, and due to the effect of the parasympathetic, the frequency will be 70 beats per minute or 70 action potentials per minute. Now what happens after the SA node generates the action potential? The action potential will leave the SA node, it will spread to the atrial muscle fibers, it will spread through the muscle fibers of both right and left atria, and then through its spread via the muscle fibers, it will reach the AV node. In the AV node, there will be some delay of the action potential for about 0.1 second, and this delay is very important. Why? Because it will allow the atria to contract first before the ventricles contract, because if they contract at the same time, the atria and the ventricle, the heart cannot pump blood efficiently into the systemic and pulmonary circulations. Why this delay happens? Because the number of gap junctions in the intercalated disc between the autorhythmic fibers is fewer in the between the autorhythmic fibers here in the AV node. Okay, then after the delay, the action potential will spread through the AV bundle or bundle of His, and then down the right and left bundle branches to the apex of the heart, then into the wall of the left ventricle and the right ventricle through the Purkinje fibers. So this is the normal path or way of action propagation from the SA node to the walls of the ventricles. So the atria contract ahead of the ventricles, and this will allow filling of the ventricles, as I said. Then both ventricles will contract simultaneously to produce effective pressure within the ventricles to pump blood into the systemic and pulmonary circulations. And that's why we need the delay at the AV node. So cardiac excitation normally begins in the sinoatrial node, located in the right wall just inferior and lateral to the opening of the superior vena cava. Each action potential from the SA node propagates through both atria via gap junctions in the intercalated discs of the atrial muscle fibers. Following the action potential, the atria will contract to empty blood into the ventricles. By conducting along the atrial muscle fibers, the action potential reaches the atrioventricular node that is located in the interatrial septum just anterior to the opening of the coronary sinus. From the AV node, after the delay in the AV node, the action potential enters the AV bundle. It is the only site where action potential can conduct from the atria to the ventricles, because elsewhere the fibrous skeleton of the heart electrically insulates the atria from the ventricles. And this is very important to have contraction of the atria before the contractions of the ventricles. Action potential then enters both the right and left bundle branches. The bundle branches extend through the interventricular septum towards the apex of the heart, and finally the large diameter Purkinje fibers conduct action potential beginning at the apex of the heart upward to the remainder of the ventricular myocardium, then the ventricles will contract. Action potential is initiated by the SA node, travels along the conduction system, and spreads out to excite the working atrial and ventricular muscle fibers that we call them the contractile fibers. I said 99% of cardiac muscle cells are contractile fibers. They are fibers that are able to contract. So now the action potential had arrived to the different contractile fibers in the atria and then in the ventricles, and the action potential that occurs in a contractile muscle fiber as follow. First, there will be depolarization similar to neurons and skeletal muscles. Then there is a plateau, which is an additional phase different from the neurons and skeletal muscles. And the last phase of action potential in the cardiac muscle cells is repolarization. Here in this figure we can see the different phases of an action potential of a ventricular contractile fiber. You can see here the rapid depolarization. It is due to the inflow of sodium, which is similar to what happens in the neurons and skeletal muscles, due to inflow when the voltage gated fast sodium channels open. Then the second phase, which is different from the neurons and skeletal muscles, is the plateau. It is maintained depolarization due to calcium influx when voltage gated slow calcium channels open, and the potassium outflow when sodium potassium channels open. So there is a balance between sodium exit, calcium entry, so we have a plateau. Then the calcium channels will open and more potassium channels will open, calcium channels will close and more potassium channels will open, so there will be loss of positive charges from the cells, so we'll have the repolarization due to closure of calcium channels, and the potassium outflow when additional voltage gated potassium channels open. So the rapid depolarization and plateau are part of depolarization. Both form the depolarization, while the third phase is the actual repolarization. An action potential in a ventricular contractile fiber takes about 0.3 seconds. The refractory period extends from the beginning of the rapid depolarization to the mid part of repolarization, and during this refractory period contraction will happen. So the depolarization phase, unlike autorhythmic fibers, contractile fibers have stable resting membrane potential that is close to minus 90 millivolt. When a contractile fiber is brought to threshold via action potential that was transmitted from conducting cell or autorhythmic cell through the gap junction, voltage gated fast sodium channels will open, sodium influx will happen. This will lead to rapid depolarization. Within few milliseconds, the fast sodium channels automatically inactivate or close, so sodium inflow will decrease. In the plateau phase period of maintained depolarization, it is due in part to the opening of voltage gated slow calcium channels, so calcium moves from the interstitial fluid into the cytosol, so more calcium to pour out of the sarcoplasmic reticulum into the cytosol. This will increase the calcium concentration in the cytosol, which is important for the contraction. Just before the plateau phase begins, some of the voltage gated potassium channels also open, so potassium leaves the contractile fiber, will balance the calcium influx. This will lead to the sustained depolarization or the flat depolarization phase. The plateau phase lasts for about 0.25 seconds. So depolarization of neuron and skeletal muscle fiber is only about 0.01 second because there is no plateau phase in the action potential of the neuron and the skeletal muscle. The repolarization phase recovery of the resting membrane potential. It resembles that in other excitable cells. After a delay, additional voltage-gated potassium channels will open. Outflow of potassium restores the negative resting membrane potential back to minus 90 millivolt. At the same time, calcium channels are closing, which contributes to repolarization. The refractory period, the time interval during which a second contraction or second action potential cannot be triggered. It lasts longer than the contraction itself, so another action potential cannot begin until relaxation is well underway. So this will help the heart to contract and pump blood efficiently and avoid the occurrence of the tetanus. Tetanus, which is maintained contraction, cannot occur due to the long refractory period in the cardiac muscles. So not like skeletal muscles, otherwise blood flow will cease. So the heart cannot pump blood properly. The mechanism of contraction is similar in cardiac and skeletal muscle. The action potential leads to mechanical response, that is contraction, after a short delay. As calcium concentration rises in the cytosol of the contractile cell, calcium binds to the regulatory protein troponin. This allows actin and myosin filaments to begin sliding past one another, and this will lead to tension development or strength of contraction. Substances that alter the movement of calcium through the slow calcium channels can influence the strength of heart contractions, like epinephrine. It will increase contraction force by enhancing calcium flow into the cytosol. Action potential and contraction. As you can see here, this is an example of ventricular muscle. We can see the rapid depolarization phase, the plateau and the repolarization, and the absolute refractory period. And during that absolute refractory period, the contraction will occur after a short delay. Here we can see the shape of action potential in the different parts of the heart. This is the pacemaker potential and the action potential in the SA node. And after short time, there is a delay between one and the other. This will allow the contraction of the atria first, followed by the ventricles. Here we can see the action potential of an atrial muscle. It has rapid depolarization, plateau phase, and repolarization. Then we see the action potential, the pacemaker, and the action potential in the AV node, autorhythmic fiber, and finally the action potential in the ventricular muscle, where we see a longer plateau because ventricular systole is long to eject efficiently blood into the systemic and pulmonary circulations. Electrocardiogram or ECG, it is an electrical recording from the surface of the body, and it records the electrical activity of the entire heart, not one cell, or it is not for one autorhythmic cell and not one contractile cells, but the electrical activity of all cells of the heart, autorhythmic cells and contractile cells at one point of time. So as action potentials propagate through the heart, they generate electrical currents that can be detected at the surface of the body or using electrodes attached to the skin. This is the shape, or this is the trace of the electrical activity that are recorded from the surface of the body, and we call this trace the ECG. So in the ECG has different components that we'll explain in details in the coming slides. The ECG is a composite record of action potentials produced by all the heart muscle fibers during each heartbeat. By comparing tracing from different leads or from different views of the heart with one another, and with the normal records it is possible to determine if the conducting pathway is abnormal, if the heart is enlarged, if certain regions of the heart are damaged, and the causes of chest pain, like what happens in angina and myocardial infarction. As you can see, the ECG has different components or different deflections. It has the baseline or the isoelectric line, and it has upward deflections like these and downward deflections. The deflections from the baseline are called waves. So we have P wave, we have Q wave, an R wave, S wave, and a T wave. 3 recognizable deflections with each heartbeat. These are the P wave, as I said. Then we have the QRS complex, the Q wave with the R wave, and the S wave. We call them the QRS complex or interval. And then we have the T wave. The P wave represents atrial depolarization. So whenever you see a P wave on an ECG, it means this is the electrical activity during atrial depolarization. QRS interval represents ventricular depolarization. So the 3 waves, the Q wave, R wave, and S wave together, called the QRS interval. The 3 of them together, they represent the entire electrical activity in both ventricles. The T wave represents ventricular repolarization. There is a correlation of the ECG waves with the atrial and ventricular systole. Systole means contraction of the heart chamber. So atrial contraction is called atrial systole, ventricular contraction is called ventricular systole. The atria and the ventricles depolarize and then they contract at different times because the conduction system routes cardiac action potentials along a specific pathway, which is the conduction system of the heart. Systole refers to the phase of contraction. Diastole refers to the phase of relaxation. The ECG waves predict the timing of atrial and ventricular systole and diastole. Again, I say predict. They do not represent. ECG measures the electrical activity, but it doesn't measure or give any indication about systole and diastole. Just we can predict when systole and diastole are occurring in an ECG. So the cardiac action potential, let's talk quickly about the different waves. Cardiac action potentials arise in the SA node, and then they propagate through the atrial muscle to reach the AV node. The propagation of these action potential, or as the atrial contractile fibers depolarize, we can see the P wave on the ECG. So the P wave represents depolarization of the atrial contractile fibers of both atria, the right and left. After the P wave begins, the atria will contract. So we can predict that during the P wave, or just short time after the start of the P wave, we can predict that there is atrial systole going on. Conduction of action potential slows at the AV node because fibers there have smaller diameter and fewer gap junctions. So the action potential will be delayed for 0.1 second before they enter the AV bundle to reach the ventricles. The action potential then propagates rapidly. As it enters the AV bundle, then it will propagate rapidly through the bundle branches and the Purkinje fibers. So the 0.2 seconds after the onset of a P wave, it has propagated through the bundle branches, Purkinje fibers, and the entire ventricular myocardium. Depolarization progresses down the septum or the interventricular septum upward from the endocardial surface, and this propagation of the action potential in the ventricular tissue will give me the QRS complex. Atrial repolarization occurs at the same time, but it is masked. Why? Because the QRS is large due to thicker walls or thicker wall of both ventricles. So atrial repolarization usually is not seen on an ECG because it is buried inside the QRS, or it occurs during the QRS, which has a large amplitude due to the thick wall of the ventricles. Then contraction of the ventricles, of the ventricular contractile fibers or ventricular systole will occur shortly after the onset of QRS complex and continues during the ST segment. The ST segment is this flat part of the isoelectric line that follows the QRS interval. As contraction proceeds from the apex towards the base of the heart, blood is squeezed upward towards the semilunar valves, the aortic valve, and the pulmonary valve. Repolarization of the ventricular contractile fibers begins at the apex and spreads throughout the ventricular myocardium, and this will lead to the appearance of the T wave, which appears about 0.4 seconds after the onset of a P wave. Shortly after the T wave begins, the ventricular or the ventricles start to relax, and this is ventricular diastole. So this part here or here, the ventricular diastole, or we can predict that ventricular diastole will start shortly after the beginning of T wave and will continue after the T wave. By 0.6 seconds, ventricular repolarization is complete and ventricular contractile fibers are relaxed. This is a summary of all the electrical events that happen in the atria, in the conduction system and ventricles in one cardiac cycle that starts with the generation of action potentials in the SA node and their propagation till the ventricular contractile muscle fibers through the conduction system. And this is how an ECG looks like in clinical life. ECG, usually we have not only one recording, but we record from different parts, or we attach the electrodes to different parts of the body, and this will give me different views of the heart. That's why the usual ECG recording is called the 12-lead ECG, which is an ECG that has 12 views to the heart from different angles, and this will give me a more 3D view of the electrical activity of the heart. ECG will be well covered in the cardiovascular module in phase 2, inshallah. Thank you very much for your patience. I will appreciate any questions by email. Thank you and goodbye.